Hello, my name is Jason Adrians. This is my presentation on mitosis. Today's 2/15/2025. Throughout this presentation, I'm going to be using the lab materials and the models from when I did the lab to describe the phases of mitosis. And this picture is representing prophase, but before the cell undergoes mitosis, something that it needs to do is go through interphase. And interphase has three stages: the G1 phase or the gap1 phase, the S phase or the synthesis phase, and the G2 phase or the gap2 phase. These are just processes that allow the cell to undergo mitosis. The gap1 phase is essentially just growth of the cell. The synthesis phase is the duplication of the DNA, and the G2 phase, the cell creates proteins that allow it to undergo mitosis properly. And then, after the G2 phase, that's when mitosis starts. And mitosis starts with prophase. And prophase is where the DNA condenses to where you can see it. And obviously the DNA is already duplicated. And the centrosomes form, and they start to migrate to opposite poles of the cell. Then the nuclear membrane also starts to break down, and it is mostly it's gone before prometaphase starts. So this brings us to prometaphase. Prometaphase, like I mentioned, the nuclear membrane has completely broken down, so there's no nucleus anymore. And this allows the centrosomes, which are now 180 degrees apart or completely on polar sides, it allows microtubules to extend, which connect to the kinetochores on the DNA, which is the middle part. And then what happens after that is connected, then the DNA starts to migrate towards the metaphase plate, which sets the stage for metaphase.